Thank you for joining us for today's webinar, A Model for Metastasis, with MSK researcher Dr. Richard White. My name is India Lachlan, and I'm a member of the Memorial Sloan Kettering Development Team. We are excited to share today's presentation as part of the spring webinar series that's hosted by our office and is available to members of the MSK community. Today's presentation will be followed by a brief Q&A featuring questions received in advance. After the webinar, the recording will be available for you to revisit and share online. Now I'm honored to introduce cancer biologist and oncologist Dr. Richard White. Dr. White and his lab colleagues at Memorial Sloan Kettering study the evolutionary mechanisms that promote tumor progression and metastasis. They use zebrafish as a model system because zebrafish have a similar genetic structure to humans. By understanding how cancer metastasizes, Dr. White and his colleagues aim to advance treatments that intervene to slow or halt cancer's progress. Dr. White earned his MD and PhD at Albany Medical College and completed his residency at Yale New Haven Hospital. His studies include 2 fellowships, one at Harvard Medical School and the other at Dana-Farber Cancer Institute and Massachusetts General Hospital. At MSK, Dr. White holds 2 appointments, one on the Gastrointestinal Oncology Service and the other in the Cancer Biology and Genetics program. Today, Dr. White will discuss his lab studies on metastasis. His goal is turning metastasis into a treatable condition. Dr. White, thank you so much for speaking with us today. We're excited to have you here.
Great. Well, thank you. I'm excited to be here and talk a little bit about our lab's work. And as India said, what I'll talk about today is this really massive problem of metastasis and how my lab and other labs are trying to go after this problem. So before I talk, I thought it might be useful to describe just in a little bit what a physician scientist is and what people like me do at Sloan Kettering and at other institutions as well. So this is, sort of a cartoon version of the 2 flip sides of my descriptor as a physician scientist. And this sort of describes how I spend my time. And that means about 2 weeks a year, I'm a typical oncologist. I see patients on the inpatient oncology side at Memorial Hospital. And for the remainder of the year, actually the bulk of the year, I run a very basic science laboratory where we use zebrafish, which I'll explain why, as a model for metastasis. And these 2 sides, although seemingly at odds with each other, actually sort of cooperate with each other, where my clinical work can actually deeply inform what I do in the laboratory, and vice versa. What we observe in the lab ultimately helps us to think about how we treat patients with advanced cancer and metastasis. So for those of you who follow how people get to this route, you sort of don't become a zebrafish physician scientist very quickly. It takes quite a long time, and this is just sort of almost a timeline of how I got to where I am, starting out in college in the late 1980s, and then eventually doing a residency, and then a chief residency, and then followed by multiple fellowships where I got my oncology training, and that's also where I gained my postdoctoral experience in learning how to do zebrafish biology and very sort of basic cancer biology, and then eventually came to Sloan Kettering for my first full-time faculty position in 2012. So as you can see, this is a very long process. Along the way, lots of things happen, including lots of great science, including the birth of my daughter, who likes to dress up, and that was very exciting. But I think it's been very rewarding because in a lot of ways, coming to understand both sides of clinical medicine, clinical oncology, but also the basic science side of it, there's no way to really rush that process, right? That really takes a long time to get to the point where you can integrate those 2 things together. So the problem that my laboratory studies is encapsulated here, which is the problem of metastasis. And in its most straightforward definition, metastasis is the process by which a cancer starts in one organ of the body and then moves to a new organ. And this is a very pervasive problem in cancer. So for instance, a cancer might start in a place like the colon or the pancreas or the skin, and then it can really jump around very aggressively to other parts of the body, like the brain or other parts of the lung and the kidney and the bone marrow. And this problem of metastasis is by far the most difficult problem that we have to face in cancer biology. And the reason for that you can sort of see here. So this is looking at all different kinds of cancers. And if we look at the survival from all kinds of cancers, you can see prostate on the top in both white patients as well as black patients, and going all the way down to the bottom with pancreas. And this is just looking at how many patients survive 5 years. And you can see there's really a huge range of survival. So you can see most patients with prostate cancer actually do quite well and survive 5 years. And if you look all the way at the bottom, obviously pancreatic cancer, most patients unfortunately do not survive survive those 5 years. But this is looking at all cancer stages combined. If we sort of look more carefully at stage 4 disease, and so this is an example of survival from pancreatic cancer, we realize that the reason why we see this huge spread is because when patients develop stage 4 or metastatic disease, that really is the determinant of how long a patient is going to survive. And this is going to be true of things like prostate cancer as well. If a patient has stage 4 prostate cancer, their survival typically is not 5 years. It's actually about 2 years or maybe 3 years. And that's true of breast cancer and a variety of other cancers as well. And I think one of the take-home points for me when I began in this field is that we can have really, really excellent survival from cancer as long as it's early stage cancer. However, once that cancer has metastasized and has spread and become stage 4, that's really where we run into trouble, and that's where the patients really don't survive very well. And so when I started my laboratory, this was really the problem we wanted to take on. Why is survival from stage 4 or metastatic cancer really so poor in most patients? And this has continued to perplex many cancer biologists over the years. So in thinking about this question of what explains this poor survival from metastatic cancer, I put up a few hypotheses here for why it is so hard to treat metastasis. One might be this observation that there are what seem to be nearly unlimited mechanisms to generate metastatic lesions. And I put the word nearly in because it can seem like that sometimes. It seems like there's so many different ways for a cancer to spread to new parts of the body. And we're seeing that now that if we start looking at tumors both within patients and between patients, there's just extreme variability, which is sometimes called heterogeneity. And this makes it seem like there are nearly unlimited ways because every time we look at a new patient, they seem to have their own way of metastasizing. I think as we get more sophisticated, we're going to realize it's not unlimited. There are a limited number of ways to do that, but we haven't figured them all out yet, so it seems like it's an unlimited way. The second big problem with metastasis is that it's what's called a whole body problem. So for many years, the way that we have most effectively studied cancer is by studying the cancer cells themselves. We isolate those cells and we study them in a dish, or we study them, in isolation, and that has led to dramatic advances in our understanding of cancer. But what it hasn't done is it hasn't led to a really deep understanding of metastasis, because metastasis by definition is the way those cancer cells interact with the rest of the body. And so of course studying those individual cancer cells in isolation doesn't solve that problem. And the third big problem is that there's very few models for studying metastasis, because as I said, most things are done in isolated cells. How do we study this really, really complicated whole body problem? And compounding this problem is that in humans, this process of metastasis typically occurs very late. Patients often present when they've had cancer for many years, and that makes it a really, really difficult problem to study in patients. So when we think about cancer, we tend to think of cancer or metastasis as an ecosystem, and I'll explain what I mean by that. So if we think about the tumor cells, and let's call those seeds, like plant seeds, those cells in isolation, just like a seed, can't do anything on its own. That seed or tumor cell only becomes really relevant when it's in the proper soil, when it's next to the other parts of the body that give it its energy and give it its resources. And so this combination, and if we use this metaphor of the seed being the tumor cell and soil being the rest of the body, has led to this hypothesis, which is actually a very old hypothesis, called the seed and soil hypothesis. And the basic idea here is that an individual tumor cell will only be able to grow and flourish when it's in the proper soil. And in metastatic terms, that means when it lands in a part of the body that gives it the resources and the energy to really take hold. And that can be in the brain or in the pancreas or in the kidney, wherever the case may be. And this phenomena, or this idea of the metastasis being a seed in soil, I think even though it was really posited a very long time ago, increasingly with more sophisticated methods is turning out to be correct, and this is turning out to be true. That metastasis is not autonomous to the cell, it depends on the proper soil, and I'll talk a bit more about that soil in a moment. So this led to the question of why, as we talked about in the beginning, my laboratory studies this problem using kind of an unusual and somewhat bizarre model for studying this called the zebrafish. So this is a video of some fish swimming around, and you can see these are fish that you might see at a very typical pet store. So each fish is about an inch long. But the fish has some really unusual characteristics that allow us to understand and really study this problem of cancer and metastasis. And I'll explain some of the reasons for this. One of the major advantages of using the zebrafish is that we were able to engineer a number of years ago a type of zebrafish that is, on the top is a regular zebrafish, that is more transparent. And so this animal is called Casper. So Casper has, and it's sort of named after, the cartoon character in a lot of ways, has some very clear advantages for us thinking about the biology of metastasis. So the first, and I think is pretty clear, is that you can see virtually all of its internal organs just with the naked eye. So you can see its brain, and you can see its intestines, and its lungs, and its kidneys, and all these things. So this is a huge advantage because, as I had mentioned earlier, one of the fundamental problems in metastasis is that it occurs late in the process. It's a late stage problem. So if we can't see it occurring, there's no way for us to know, why did that cell go to this part of the body? Why did this cell go to a different part of the body? And part of the real challenge in studying metastases over these years has been to visualize this and really capture those events at a very early stage. And so when we created this technology, this really gave us a window into metastasis that really other models that have been used were not able to achieve. The other question that often comes up, though, is, yeah, but this is still a fish, right? How related is a fish to a human? And I think one of the other really surprising results that came out of a lot of our work and many, many generations of scientists is that obviously fish and humans don't look at all like each other, but really surprisingly, we share the vast majority of our genes. So humans have about 20,000 genes, fish have a couple more, but we share 70% of our genes at the level of DNA. And this is really a shocking, revelation for a lot of us, is that even though these animals look so different, they share the fundamental genetic machinery, or DNA, between humans and fish, and it turns out mice and lots of other animals. And this allows us to then make inferences about what happens in a fish and how that then applies to humans. So it's a really powerful model for allowing us to study things like genetics and DNA, but now in an animal that's transparent such that we can see metastases and visualize metastases as they're moving around the body. And I'm going to show one example here of how we visualize these metastases. So the video I'm about to show is an example of a Casper animal, and you'll see the tumor cells are labeled in a fluorescent green protein, and we can actually watch this occur. So that's where the tumor starts. And you can see over the period of several weeks, the tumors spread into new parts of the body, moving towards the head, and then we have animals like this. And this level of resolution is really unprecedented in almost any animal. This allows us to really capture how metastasis is happening in real time. And one of the real advantages of the zebrafish, because it is so transparent, is we can now take animals like this and study them with very, very sophisticated microscopes that let us now hone in on really exactly what's happening to the first cancer cell as it arrives at a new location. And so to do that, we use an animal that looks like this. So this is a highly zoomed in picture of what's happening, similar to what I just showed in the previous video, where the tumor cells here are green and the red cells here are blood vessels, because we know that's how tumor cells get around the body. They move around using blood vessels. So when it moves from one part of the body to another part of the body, it has to pass through the bloodstream. And so what we're capturing in this video here is that green cell right in the middle of the video. You'll see that is the very first cell that's metastasizing and moving through the bloodstream, and you see it's kind of stuck in a blood vessel, and that cell is about to exit the blood vessel, and it's about to do it, and right there it is. And that cell has now jumped out of a blood vessel and is now sitting in a new tissue in the body. And this is really one of the first examples where we can very rapidly and easily visualize what happens in those very first moments when a cell arrives and metastasizes to a new place in the body. And this gives us a really striking opportunity to figure out, well, what is it about that cancer cell that makes it special? And more importantly, what is it about the surrounding cells that allow it to take hold in that spot? And those are really the questions that we're incredibly interested in. So as I mentioned earlier when I was talking about the seed and soil hypothesis, is that this is an ecosystem, and this soil is sometimes called the tumor microenvironment because as we're showing on the top, now we can see the tumor cells quite readily. But the question that we and many others in this field are trying to get at is, well, what makes up the soil? What is part of what is sometimes called the microenvironment? And I listed a few cell types here that make up part of that microenvironment. And these can be cells like adipocytes or fat cells and fibroblasts, lymphocytes, which I'll talk a bit more about in a second, and all these other cell types. And this is a really complicated problem because although the tumor cells are obviously the most important part of how a cancer grows, they get nurtured and supported by all these other cell types and these sort of bidirectional interactions between the tumor cell and all these other cell types. And this is a really difficult problem to study, but I think increasing evidence would suggest that it's not just one of these cells in the soil or microenvironment that support the tumor cells, it's really a concerted activity of all of these cell types that really allow the tumor cells to grow. And I think this diagram really helps to explain why metastasis is such a complex problem, because we're not targeting just one type of thing, we're actually targeting many different interactions. So how important is the microenvironment or this soil idea? And I think the best evidence for how really remarkably important the soil or microenvironment is comes from really the dramatic results that we're beginning to see both here at Sloan Kettering, but also really throughout the world with the success of a type of therapy called immunotherapy. And the way immunotherapy works is it was long ago recognized that one of the cells in the soil or the microenvironment are lymphocytes or immune cells. And it was actually a very long-standing question. If cancer cells are really abnormal, why can't your immune system recognize them and get rid of them? And I think over years it was recognized, well, sometimes the immune system can do that. And so through a series of really groundbreaking work, both done here as well as many other institutions, it was recognized that the problem was that the tumor cells were actually suppressing those immune cells. We're suppressing a type of immune cell called a lymphocyte, and specifically a T lymphocyte. And with that recognition that those tumor cells were sort of damping down that immune response, it became sort of clear that one way of maybe going after cancer would be to reawaken those lymphocytes, turn those T cells or those T lymphocytes back on and allow the immune system to target those cancer cells. And that has really led to a remarkable transformation of certain diseases. So on the right hand, this is the most recent data looking at survival from a disease called melanoma, which is a type of skin cancer, looking at how we do with immunotherapy. And if you look at the very top graph, which is this orange one that says nivolumab plus ipilimumab, that's a combination immunotherapy that activates those lymphocytes and then dramatically improves survival of melanoma. If we looked at a graph like this, say 20 years ago, the vast majority of patients would have succumbed to their disease by this point. But now with the advent of these really sophisticated immunotherapies, we get really remarkable survival from this disease. And I think for me and many others, this has been a really exciting observation because in this example, we're not really targeting the tumor cell. What we're really targeting is the microenvironment or the soil. And of course that interacts with the tumor cell, but I think that this one piece of data really has led to a tremendous amount of excitement that maybe one way of improving survival from cancer or from metastasis is to go after the microenvironment or the soil. So what my laboratory has been doing is now broadening this to look at many other cells in the microenvironment, because as I said earlier, immune cells are one important part of the microenvironment, but there are tons of other cell types in the microenvironment. And so we became interested in a question of when a cancer cell arrives at a new location, how does it get its energy, right? Because for a cell to divide, that takes a lot of energy. It needs lots of nutrients, it needs blood vessels and lots of sugar and fat and all those other things in order for it to grow. And this has been really a burgeoning area for a lot of people to think about how does a cell get its nutrition? And one of the things that we've been doing, and you can see here on the left, this is a picture of a zebrafish in which we were looking at a tumor, again in green, and we started looking at the components of those tumor cells, and that's the zoomed in image below. And if you look at sort of the orange cells, you can see those cells are stained with fat or lipids. And lipids, in addition to doing many things in the body, are at the most straightforward level, they're fuel. that almost every cell in the body in one form or another can use lipids and turn that into energy. And so we had this hypothesis that maybe these lipids in these tumor cells are being used for fuel in the cancer. And that in turn led us to ask, well, if they are using these lipids, where are they getting them from? And on the image on the right, you can see this is an image of one of our animals where you can see the tumor cells, in this case a melanoma, are all in purple there. And all those white cells that are surrounding and sort of interspersed with the tumor, those are all a cell type called an adipocyte, or a fat cell. And in work that we subsequently discovered, it turns out that those fat cells can directly donate their energy store, their lipids or fats, directly into the tumor cell. And so what happens is that you have an adipocyte sitting in the soil. It can then release its fat into the microenvironment. That can then be taken up by a tumor cell like a melanoma cell. It can then, and the way it does this through a specific series of proteins called the FAT-P proteins. And once it's inside the melanoma cell can then use this energy or this lipid or fat to really proliferate and grow. And very excitingly, we and others are beginning to identify molecules that can block this process, that can stop that from occurring. And that's still ongoing work. But the logic is that eventually we should be able to block the ability of the tumor cell to take up these nutrients. Another example, I think, where the environment provides something to the tumor cell, in this case it happens to be a fat cell, but I think ultimately this will represent a really new and exciting therapeutic target. And I imagine as we go on, we're going to discover lots of other new cell types in the microenvironment or the soil that I think will help the tumor grow and that will represent new targets in cancer. So this leads and has led lots of us to ask a question of, can we find drugs that target the energy source of the tumor cell? And there's a lot of excitement in this field, and this is a challenging process. I think as many of you in the audience know, research takes a long time. I sort of alluded to this at the very beginning, and I sort of lay out some of the things that are involved in turning a basic science observation like we make in the zebrafish into a potential drug, finding the right target or gene in our case. That's a pretty solid 5 years of lab work. We try and work quicker, but it does take time. How do you find the right drug? That takes another couple of years. How do we find the right patients? We now know we don't generally try to just blindly treat patients. We now do genetic testing on the tumors to try and figure out which patients are more likely to respond or less likely to respond, such that we really target the right patients. At MSK, we do a genetic test called impact testing, and there are other tests at other institutions. And this has revolutionized the way we stratify or pick the patients for the best trial. And then ultimately, we go on and do clinical trials. And these are phase 1, phase 2, phase 3 trials. And these obviously all take time. But I think when you sort of lay out, it becomes clear why it takes sometimes some time for us to get there, but very often it starts with a very basic observation that then leads to really this explosion of therapies, just like we've seen for immunotherapy. And we're very hopeful that we'll see this in lots of other targets outside of the immune system. So I started out by saying there seems to be nearly unlimited ways for cells to metastasize, but I think as we think about it more carefully, my view is that the problem of metastasis is ultimately solvable, but solving that problem will require us to consider it as an ecosystem, where we really need to understand the specific ways that tumor cells interact with all the different things in the rest of the body, all the different cell types and proteins, and really treat this as an ecosystem problem. And on the bottom right there, I think there's no one perfect way to solve this problem. It's very clear that we have to study human patients and tissues that come from their patients and the genetics of the tumor. But then we also need really powerful models like the zebrafish, mice, which is a very common model, and probably other models as well. And it really is this idea of a collaborative effort between all these different systems that will ultimately, in my view, turn metastasis into a solvable problem with this sort of view of it as an ecosystem problem. And the last thing that I think is very important to acknowledge is the really huge number of people that have contributed to this work, including members of my laboratory who've done really the vast majority of the experiments that I talked about. We have a growing team that works on this and people who were in my lab before. But on the right, I list a very large number of collaborators, and these are people both within Sloan Kettering, but also outside of Sloan Kettering as well, because I think it's increasingly clear that cancer and metastasis in particular has so many dimensions that it's almost impossible for one lab or one person to study all these dimensions. And so we really rely on these highly collaborative efforts to try and solve this problem and bring together teams with very, very different skills and abilities to really try and tackle this problem. So with that, I would like to thank everyone for being here, and I'm happy to take any questions.
Great. Thank you so much, Dr. White. This has been fascinating. During registration, we gave participants the opportunity to ask questions, and we received far more than our time permits today. Thank you to everyone for sharing these questions. Many of them that we received were related to specific diagnoses, and we encourage those individuals to discuss their questions with their physicians. So now we have time for a few questions, and I'll jump right in with the first one. Are certain cancers more prone to metastasis, or is it common across all cancers?
I think it seems to be common to virtually all cancers. And the answer seems to be that stage plays a big difference. How large is the tumor seems to play a very big role in that. So obviously a much smaller tumor has a much lower chance of metastasizing, and the bigger the tumor gets, the higher the chance. If we look long enough, most tumors, whether that's prostate or pancreatic cancer, have the capacity for metastasis. I think what we're increasingly recognizing is that the ability for metastasis is also determined in part due to the genetics of the tumor, like what gene mutations does it have, but also what's called the epigenetics of the tumor. This is not so much the DNA sequence, but the way the genes get turned on and off. So our answer seems to be that most cancers can do it, but some cancers do it much more rapidly than others. Pancreatic cancer being one very dramatic example of a tumor that likely does metastasize relatively easily, and others like prostate where it takes more for that to happen.
The next one is a two-part question. What are the most promising research opportunities in metastasis, and are there any new treatment options on the horizon?
So I think I'll answer the first part. I think that there really are two threads that have been running through the field right now. One is the idea of molecularly targeted therapies. So this is, we find a mutation in the tumor itself that makes it susceptible to a particular type of drug. We've seen very dramatic examples of that in melanoma, lung cancer with so-called targeted therapies. And in concert with that is now the explosion of immunotherapy, so the idea of treating the surrounding of the tumor or the environment of the tumor. And really, there's a huge amount of energy right now in combining those two approaches. How do we sequence those, put those things together such that we can target the tumor cell itself with a targeted therapy and then the environment or the microenvironment of the tumor with an immunotherapy? I think this is a tremendously exciting area. There's already hints that this is likely to work, these combinations. And then I think beyond that, my guess will be, as we identify newer and newer targets in the microenvironment, those can then be layered on to these types of therapies. So for example, I suspect that targeting the adipocytes like I showed would be one such idea, and I think that's a big opportunity for growth.
Great. How does your work apply to different cancer types? I know you'd mentioned melanoma, but how does it relate to others?
Yeah, I think one of the things that my lab and I think other labs at Sloan Kettering are interested in doing is finding out the principles that are common across different tumor types. So we've used melanoma because we were able to generate these and we learned a lot of the basic biology, but now we're at a point where we want to know if some of the principles that we've seen in melanoma are really more broad and do they apply to other cancers. So for example, we now have a project in my lab looking at pancreatic cancer because we think this is a huge clinical problem. It's a really unmet need. But we imagine a lot of the observations that we're finding in melanoma and pancreatic cancer are going to apply to those other cancers. And we're already beginning to see examples of that. When immunotherapy came onto the horizon, it was really limited at the beginning to melanoma and kidney cancer, but now we're seeing really dramatic effects with other types of cancers like lung cancer, and I imagine that's going to continue to be a theme, that the underlying principles are going to be broadly applicable to many cancer types.
And the last question, have there been any advances in the early detection of metastasis?
Yeah, I think this is a really rapidly growing and very quickly moving area. As I had mentioned earlier, one of the things that we see is that cancer cells have to move through blood vessels to get to their new locations. And so this has led to this real explosion of the idea of, can we detect those cells or pieces of those cells long before we could ever see them, either by the eye or if the patient develops a metastasis. And this has led to this two fields, one called circulating tumor cells, where we can capture those cells using certain types of machines, and more recently, the idea of circulating free tumor DNA, where we can capture snippets of the DNA that get released from the tumor cell and now float in circulation. And I think there's already been really, really exciting advances in both of those, that we can capture both the cells as well as the snippets of those cells very, very early, long before we could ever see the cancer. And I think one really exciting application of this is that we may be able to then predict who or which patients are likely to relapse and maybe treat them ahead of time before that relapse occurs. So this is a very quickly moving area, but I think a very exciting one.
Well, Dr. White, thank you so much for your time today, and thank you to everyone who joined us. We hope you found the presentation valuable, and if you're interested in learning more about our research, we encourage you to keep an eye out for emails and other updates from MSK. Finally, you'll receive a follow-up email about this webinar linking you to a survey about your experience, and we hope you'll take the time to give us your feedback so that we can continue to bring you useful content. Thanks once again for joining us, and have a wonderful afternoon.